Hello, my name's Steve Werblow. I'm president of IFAJ, and it's an absolute pleasure to kick off today's webinar series, which we're doing in conjunction with Syngenta. So Syngenta stepped forward and helping us do a deep dive into the topics that we as journalists cover, which is really exciting. This is part of our professional development effort, first step towards a new wave of education programs. And what you're going to find is it's not a commercial. This is an opportunity for us to hear from scientists who are doing the work that we report on. And so we encourage you to listen, to share with your colleagues, and to ask great questions. We're going to hear these presentations. We're going to have about 15 minutes at the end for questions. For those questions, at the bottom of your screen, you'll see the Q&A button. Click that button, type in your question. If you see a question that you are particularly excited about, you can upvote that question so it gets moved up the queue and has a greater likelihood of being asked and being addressed, which is great. So we're very honored today to have Dr. Yuri Krieger, Global Head of R&D for vegetable seeds and flowers for Syngenta, and Dr. Ian Jepson, who's the technical leader for the field crop seeds group at Syngenta. So again, I'm going to get out of their way. I'm going to encourage you to listen and ask great questions. And we're going to record this and post it in our webinar section of our website. So please do share with your friends. And with that, I hand it over to Dr. Krieger.
Thank you, Steve, and thank you, Martina, for the opportunity, and welcome everyone. Good morning, and I guess good afternoon to all of you. I'm going to start with a short introduction about myself. I lead the R&D organization for vegetables and flowers at Syngenta. I've been with the company about 5 years. Prior to that, I was with a company called Monsanto, later on turned into Bayer for the past for about 10 years. Well, without further ado, I'm going to start now, and Ian will continue. We have our presentation split into two, so that will allow Ian also the time to introduce himself. So today, basically, I'm going to be talking about a little bit about plant breeding. What does that mean? We hear a lot about it. There's different ideas of all the activities that actually take place in plant breeding. So we would like to shed a little bit more light on the activities that we are doing, starting with the history, a little bit of history of agriculture and crop domestication, then the foundations of the genetic studies. Don't worry, it's not intended to be a college course or by any means. Then some of the plant breeding principles, and ultimately talk a little bit about genetic diversity, and then the modern techniques in plant breeding, which will be a nice segue also to Ian's presentation, where he's going to be talking about other technologies that we're using in plant breeding, like GMO and genome editing technologies. So let's talk a little bit about the history, right? I mean, we're going to go back in time about 10,000 years ago to the first evidence that we see, probably some of the evidence that you're going even prior to that. When from in the area of the Fertile Crescent, where I'm originally from. I'm from Israel, so this all speaks very. I feel very connected to those ancient times as well, from a historical perspective, where we start seeing the first evidence of agricultural and crop domestication of wheat, barley, chickpeas, lentils, all these good stuff that has been available in the in nature. And humans, they start to basically become farmers and start using what was available in nature for their own consumption. Over that period of time of really 10,000 years, different crops in nature expressed a wide range of what we call phenotypic traits. These are traits that we can observe, we can measure them later on. And some of those traits were beneficial for humans. And slowly, the human starts to select for those particular traits. I always say that the first plant breeder was the one that actually decided that this good and healthy seed should be preserved for next year instead of consuming it or using it in the current year. And what we start seeing is that some of those traits that were beneficial for humans were also kind of locked into the next generations. You can see a much if it's bigger yield, or I'm going to talk a little bit later about these type of traits, and they really help with what we call crop domestication. So the crop that you see today in the wild, in some cases, they are even not closely remote to what you know or what you're consuming by buying it on the market or in a supermarket. And those were key drivers also for ancient cultures. If you think about the Mayas in Mexico with corn, but also the entire population, in the Mesopotamia and the Middle East in the old days, all the way to the Niles, to the Egyptians, it was crop domestication was a real driver for these cultures to thrive. So over the years, and now we're moving quite fast to the 1800s, crops selected for continue to be selected for favorable traits through the process of domestication. You can see traits like reduced toxicity, increased yield, obviously, improved taste, color, quality, etc. And later on, since the late 1800s, we start seeing more focus on mechanization. So adjust the crop plant architecture towards domestication and different ways of cultivations, using a mechanization on the farm, if it's through animals at the beginning, but later on, obviously, through more advanced technologies. And what happened was that these natural variation, those crops, those varieties that haven't been domesticated, were left behind in nature, and it was good. It's important to maintain that diversity. I'm going to touch on that in my next couple of slides. And then along these lines, in the 1800s, what also happened is the entire scientific era, kind of start to evolve, and it evolved in different areas, right? In mathematics, in physics, in microbiology. Almost every area that you can think of has been and made tremendous leaps during the 1800s and obviously later on the 1900s. And among all the great scientists, there were two that really spent a lot of time in the field of genetics, or what became later on the field of genetics. And I wanted just to speak real quick about them, because both of them, Gregor Mendel and Charles Darwin, despite that they were not related to plant breeding, their finding and their research work tremendously influenced what the plant breeding as we know them today, and the work that we are, the laws and the methods that we are using today. So Mendel basically, in a nutshell, he was a very good scientist, a great scientist, but never received that credit during his lifetime. Basically, he was looking at a lot of segregation of traits in nature, with one big exception compared to Charles Darwin. He was looking at traits that were dominated or dictated by a single gene. So every gene has two alleles, recessive and dominant. And he did a lot of his work in peas, and he was basically able to show that those genes segregate in late once you start selfing, and once you start crossing this type of a phenotype, the next generation became more predictable on what's the phenotype is going to be because of the laws that Mendel said. Charles Darwin, on the other hand, I don't know what was his relationship with Mendel. I believe that if it was today, they were not very good, because he didn't look at it from a single gene perspective. He looked at it through course in time and the evolution in this case, the different finches that he discovered in the Galapagos during his excursions. When he said, well, it's the same bird, but for some reason they have been evolved to eat, for instance, to rely on different source of nutrition, if it's seeds, if it's insects, or if it's fruit, but they all come from the same bird. So he was looking at it more holistically, that there are different genes that are driving those phenotypes, and that's where he basically established the entire theory of evolution. Now, how does that connect actually to plant breeding? When you think about what we are trying to do, and now we're moving in time to the 1900s, based on those theories, first of all, that genes are segregating independently, like Mendel said, but also that we can see small progress and differentiation based on Darwin theory of evolution. The later discussions and research was focusing on what are those genes, because they didn't know that those genes exist, but they knew that there's something hidden inside that basically define the rules of what we call heredity, how passing of physical characteristics or traits through those genes is moving from one parent to its offspring. In the early 1900s, they start developing and start observing that when you make a cross between two lines, you start seeing a phenomenon where the hybrid, those progeny of those two parents, is becoming way stronger, much better on yield, much better on vigor, and basically the correlate, the hybrid vigor or heterosis, which is became like the holy grail for those plant breeders, to find that unique match between two parents. And that truly transformed the way plant breeding is being done. First of all, it was moved from just picking the best grain that you see in a certain season and saving it for next year, to be more directed towards creating combinations of different parents. We call it a female and a male, although a corn plant or a tomato plant has both sex on that same plant. So, and if you can see what happened here, and just gonna look here into this graph. Let's see where my pointer is. Early 1800s, all the way to the 1920s, it was just a classic selection. I'm going to pick the best plant that I see in the field. I'm going to save it for next year. Then they started to make crosses between those two very good plants until about the 1950s, where they start to really create different pools of different genetic pools to truly try to maximize that heterotic effect I was talking about before. So, and why is it important? First of all, you can see the tremendous yield increase. This is ton per hectare that occurred once from around the 1950s, where seed companies like Syngenta start to create and invest more time in developing a better parents that will create the best hybrid for the farmers to plant. And you see the increase in yield has been quite dramatically since they start using this hybrid method. By the way, this is just an example from corn. It's a well-known one, but the same platform, the same way of working, the same method could be applied almost to any crop as we know it today. And then in the 2000, remember this slide, this is where the biotechnology, that's another leap, another jump that is more attributed to the biotechnology era that Ian is going to talk about it. And the reason why it's important for us to understand it, and I'm going to touch here on the last bullet point, there is a lot of, I would say, resistance in some cases to the breeding or to the seed companies, where people are saying, you know, we hear that again and again, you guys force us to buy your hybrid seeds every year, so you know you can make more money, you can increase your revenue. The fact is, though, this shift from hybrid, just selection of crossing two good lines, into developing a structured breeding program required extensive trials, obviously, and thousands of combinations that we need to check. But also, think about the Mendel law in that hybrid that you sell to the farmer. If they save that seed, obviously there's going to be some segregation of those genes in the following generation, because it went through another process of meiosis, another process of basically self-pollination. So just like the peas, the peas of Mendel segregated to yellow and green and shrink and round peas, that's basically what you're going to find in the field. And in many ways, this is the old practices over here of just saving seeds. So if you are after increased yield, you got to start thinking about, you know, maximizing the heterotic effect, which is what basically the seed companies are doing. Going to talk a little bit about genetic diversity. Remember, we left a lot of that genetic diversity out in the wild. And why it's so important? Genetic diversity is crucial. This is the gene bank, all the genes that exist basically in that particular species. Sometimes through the course of domestication, we got rid of some of the genes, because breeding is simple. You get what you select for. So if you select for high yield, you may didn't add the quality. Many of you are saying, I'm sure, oh, the old tomatoes were way, way better than the current one. Well, it depends on the market. If the market is focusing on yield, and that's what the grower is being paid for, and not quality, so the shift towards the varieties will be more towards that direction. I want to show you this beautiful example, which is one of my favorite. For instance, wild mustard, that none of us are actually consuming or you know, in our day-to-day life. But through the power of selection and plant breeding, you know, the breeders selected for different type of mutations in some cases, or phenotype that they thought would be valuable to consumer. And that's how you got the famous Romanesco types, cauliflowers that are white, that are not yellow, even purple cauliflower. Broccoli came exactly from the same plant, just selecting for different traits. You have cabbage that was selected, you know, for certain traits, you know, with that inhibit flowering, so you get that curd of leaves. A well-known one is also Brussels sprouts. You have the Chinese cabbage, even radish. They all came from the same ancestor, wild mustard, thanks to plant breeding activities. Last two examples, you know, kale, which become kind of one of the latest superfood. And this is a new innovation coming from Syngenta about 2 years ago, which is a small cauliflower that you can basically, it's branches of cauliflowers, very tasty, very sweet, that we are very proud to have in our portfolio. But aside from plant architecture, there's also resistant traits that are coming from the wild. And all those became like the toolbox of the breeder to start selecting from in order to make the best product. We often talk about mutations. Mutation breeding is very important. Mutations are changes in the genetic code, and they happen spontaneously in nature. You just saw some of them, in my previous slide. But because of the big value that it brings from a diversity perspective, we started using them also to induce them in artificially, and create through that some genetic diversity. Remember that when Ian is going to talk about GMO, diversity is key. This is the source, this is the fuel for every effective breeding program. And it can cause phenotypic changes. Sometimes they're more radical, sometimes they're even silent. But again, the true value for us is that it allows us to look for changes in the genome that will possibly address certain changes in the market or in the environment that we're facing. This is one classic example. It's a disease resistant to a virus. The entire tomato market over the past 6-7 years have been impacted through this virus. The unique thing about this virus is that it affects the fruits. It doesn't affect the leaves. So if you have it in the fruits, the entire greenhouse basically is not going to be marketable. And we see constant pressure of this type of increase in pathogens and new pathogens like viruses or bacteria, for instance, or fungi, through just the way we live today. There is more invest in crop production in key areas. We have restrictions on chemical applications. There is an increased demand, all of us, all over the world, for higher food quality. Product transportation definitely doesn't help us remove the diseases from one country to another. And on top of everything, the climate change, which is evident to impact to accelerate the emergence of new pathogens. And all of that is becoming the breeder's task to start to combat that and find solutions for the farmers so they can continue to grow the food that we need. Last slide, is just to recall, to remind everyone that it takes much more than that. And as Jeff was saying, data, Jeff Rowe, our CEO, data is the new tractor. And for us, data is becoming the equipment for us to start dealing with all these variables that are changing. We look at the different genetic combinations that we need to create. It's all about the data. We're looking about environmental data, crop management data, genetics. There's tons and different layers of data that we can take into account and towards managing the best decision on what product to select and which product to put on the market. And for that, we really using analytic and data science is becoming our core tool for our breeders and for our company in order to select the best product for our customers. So plant breeding, in a nutshell, is a combination. It's a combination of art, like you saw on the brassica and the mustard. Depends on creating some new crops and new products for us as consumers. But there's tons of science behind it that we got to take into account with the current ever-changing environments that we're facing. And with that, I'm going to move to Ian. I'm going to stop sharing, Ian, and let you share your slides.
Thank you, Yuri. Let's get back to that. Okay. So a quick introduction. My name is Ian Jepson. I'm the site head of our flagship R&D site in Research Triangle Park, North Carolina. And we have around 500 scientists working on some of the projects that I'm going to describe and some of the work that Yuri's team are doing. In addition to that, I'm a technology lead for seed research, and that means my programs are developing new technologies like genome editing, technologies to support the generation of GMOs, also genotyping and genomics technologies to support some of the breeding efforts that Yuri just described. I have a molecular biology degree. I'm a biotechnologist, and I've been with the company 35 years. So what I'd like to do today is to share with you two modern breeding technologies that really complement some of the traditional breeding that Yuri has described to you. And those technologies are, the first is genetic modified organisms, or GMO technology, and the second is genome editing technology. So these technologies are really an extension of the plant breeding principles that Yuri's just outlined. That is creating useful genetic variation to create new phenotypes. So plant breeders have been doing this for thousands of years. They've been searching for that variation in natural populations, or sometimes inducing that variation using radiation or chemicals, but that's a random process. And through that, breeders have made huge advances in disease resistance, drought tolerance, yields, flavors, and colors. So some of the things that Yuri's just shared with you. The big challenge, though, is what happens if there's no natural variation in the population. And one really good example is insect resistance. Many crops do not have the variation in their genomes to protect themselves from major pests. It's common for a lot of the vegetables. It's certainly common for a lot of the major field crops. So I'm going to describe how GMO technology and genome editing can create that variation when that natural variation does not occur in populations today. So I'd just like to pause for a second and just contrast GMO technology and genome editing. So GMO is a technology where we take a gene from another species. That could be another plant species which is not sexually compatible with the crop, or it could be from a bacteria or another microbe. So we're actually transferring a whole gene and a whole new attribute into the crop. Genome editing, on the other hand, modifies a gene that is already in the genome. So the gene may be there, it just may not work very well, and genome editing can go in and optimize that. So why do we need these new breeding technologies like GMO and genome editing? Well, the United Nations have estimated that the world's population will be 9.8 billion by 2050. And it's very sobering to think that the world's population is increasing at a rate of 200,000 people every day. And I think that's very sobering when we think we have to feed those new people with safe and sustainable methods. And that's really where these step change technologies come in. You saw Yuri's graphic where there was an inflection of biotech, and that's what we're referring to here. The techniques of GMO and genome editing created an inflection of yield. So let's talk about how we make a GMO crop. And this stems back from the work of a scientist called Dr. Mary Dell Chilton. And Mary Dell, actually, she founded our research site in 1982. And she was researching a soil bacteria called Agrobacterium tumefaciens. And this bacteria causes galls or crown galls on plants. And it does that by injecting its DNA into the plant cell and causing a tumor. So Mary Dell and other scientists figure out that mechanism, and they replace the tumor genes with genes of interest, for example, insect resistance. So in this graphic, you see the Agrobacterium on the right, the brown structure. That's a bacteria containing a circle of DNA called a TI plasmid. And into that, they've inserted a new gene. So that gene could be an insect resistance gene, for example. You then mix those bacteria, those agrobacterium, with plant cells, and the agrobacterium will infect those cells and integrate the orange DNA into the genome of the crop. You can then regenerate that plant, and all the seed from that plant thereon will contain that additional gene. So you've added a whole new gene to the crop. So let's look at some examples of that. Let's follow on. Let's keep on the track of insect resistance. So the background to this is over 100 years ago, scientists had discovered that a soil bacteria called Bacillus thuringiensis, often called BT, could selectively kill certain insect pests. And it does that because they've evolved some proteins called cryproteins or crystal proteins. And those proteins can bind to the midgut of the insect, form a pore, and kill the insect. And the beneficial nature of this was actually picked up on by the organic movement. And because of the very good human environmental safety profile of BT proteins, the organic movement have been using sprayable BT. So they culture the bacteria and then spray those spores on the field for decades. So this has been practiced for almost 100 years now. So biotechnologists picked up on that and went and cloned the genes for the BTs. They used the agrobacterium process to introduce those genes into the crop. And if we look at the example on the right here, we see corn plants that have been transformed with a BT called Cry3. The plant on the left here, this sad-looking stick-like structure, has no BT gene. When it is infested with corn rootworm, the larvae completely chew off those roots. And that means that plant can't take up water or nutrients. In fact, that plant will lodge, it will fall over. That plant will produce no yield whatsoever. The plant on the right, however, has had the BT gene that protects the corn from corn rootworm beetles. So this technology is now widely used across the United States to protect against corn rootworm. And corn rootworm causes around $1 billion of damage a year to the corn crop. So this was a pretty dramatic improvement in corn yields due to that one gene. The second example of insect resistance I want to share with you, and this is even more, this is a global issue. That is this insect here, fall armyworm. Fall armyworm originated in the Americas, but in 2017 it was detected in many African countries. 2018 it went to India. By 2019 it has spread across many Asian countries. And this year it was reported in European countries. So this pest has become a huge problem because it's in over 80 countries now. And chemical control does not control fall armyworm. What happens is it burrows into the stalk, and if you spray the chemicals on, they don't kill the larvae effectively. The only technology that can protect from fall armyworm in an effective way are GMOs. In fact, the best technology on the market is a gene called VIP3A. It's again from Bacillus thuringiensis, and it protects corn and soybeans from fall armyworm. So without that GMO technology, we would see huge crop losses across the world. The second example of a GM technology I wanted to share with you is Enogen corn for animal feed. And Enogen seeds contain an optimized, they contain a gene expressing an optimized alpha amylase. And an alpha amylase is an enzyme which breaks down starch to sugar. And if you feed that corn, the Enogen corn, to beef and dairy cattle, you see a 5 to 6% feed conversion efficiency. So that grain is easier for the cattle to digest. Therefore, you're reducing the amount of food required by those cattle. And actually, just last week, on the 14th of November, Syngenta, McDonald's, and Lopez Foods announced a major collaboration to utilize Enogen technology to produce beef in a more sustainable way. So this is an advantage to the farmer because it saves on grain and money, but it's a huge sustainability benefit when you consider the scale of beef and dairy cattle in the United States. Just to put that sustainability benefit into context, if you consider a herd of 1,000 lactating cows, you feed them on Enogen grain, and the expectations you'll see at least a 5% feed conversion efficiency. That leads to major reductions in CO2, a 1.2 million kilogram reduction in CO2 emissions on an annual basis, but also major reductions in land use, water use, and energy use. So this is a really powerful technology, not only to increase agricultural productivity, but also to make it more sustainable. Now, GM, just a comment about the safety of GM crops, and this often comes up. And what I would say is that it actually takes over 13 years to develop a new biotech GMO trait, and typically costs over $136 million. Much of that $136 million goes into product safety testing, where the developer of the technology has to show there are no environmental negative impacts. We have to show that those traits have no impact on the crop agronomy, that the genes in those crops do not impact non-target organisms. So, for example, I mentioned the BTs were selective. The corn rootworm genes only killed corn rootworm. They would not impact earthworms, bumblebees, or any beneficial insects. We have to show our crops not become weedy pests and have no impact on other biodiversity in the environment. Similarly, on the human safety side, the submitter of the regulatory package has to show that the GM proteins are not toxic and not allergenic, and they do not impact the crop nutrition. And just to summarize, many hundreds of papers discussing the safety of GM crops. One commentator made reference to the fact there have been over 3 trillion meals served containing GM crops, and not a single case of ill effects to humans. Because of the benefits of GM, I just articulated a couple here. GM crops were first launched in corn in 1996. In fact, the first insect resistance event came from our site. And you see a very rapid adoption of genetic modified crops in the large acre crops like corn, soybeans, and cotton. So that was a huge inflection where farmers adopted the technology. And if you look across the US now, those major crops, between 85 and 95% of them are now GM because of the benefits of the technology bringing to the crop, that new variation. But GMOs are not restricted to the United States. So of the acres on the market today, so there's 73 million hectares of GM in the United States, but there are 206 million hectares grown worldwide. So GMOs are a worldwide phenomenon. They are approved in 28 countries. There are more than 10 GM crops on the market today. Now, I will say, because of the regulatory costs, they typically are restricted to the broadacre field crops like corn, soybeans, cotton, and canola. So some of the smaller crops, while they are on the market, it is challenging for them to afford the cost of development of GM technology. So with that in mind, let's move to the second technology I want to share with you today, and that's genome editing. And genome editing technology, differs from GMO in that it modifies a gene that is already in the crop. And the way we do that is use a technology referred to as CRISPR-Cas technology. And this is a 2-component system. The Cas are a little pair of molecular scissors. The CRISPR is like an address on an envelope. It will tell those molecular scissors where in the genome to go and make a cut. So you design your CRISPR molecule, it will go and it will go to the chromosome and to the gene, and the location of the gene you desire, make a cut. And when that DNA is cut, it gets repaired. And that repair can be error prone, and it can cause a knockout. So you can lose gene function, or like in the graphic, we're actually writing in a new base pair. So you can correct something or optimize something. So you can knock it out or improve it. And let me share one example with you, and that is tomato texture. So typically, tomato varieties last between 10 and 12 days. In the United States and actually globally, there is huge wastage in the fresh tomato market because of delays in picking, damage in transportation, or just being delays of being sold in the grocery store. So there's a huge amount of wastage. So we launched a project to extend the shelf life of tomato quality and reduce waste. And our idea was to increase shelf life by 3 to 5 days. So we know that cell wall degradation controls the rate of ripening, and we know the genes that regulate that process. One of them is referred to as pectate lyase. So we used a CRISPR-Cas12 technology to knock out the pectate lyase gene. So we sent those molecular scissors to make a cut in the pectate lyase gene and knock it out. So the fruit still ripens, but it ripens more slowly. And we've added around 5 days onto the shelf life. So you see the edited, non-edited line on the left, it's gone all squishy. The edited line on the right, it's extended its shelf life. Now, if we now want to take this technology to market, if you go to a number of regulatory jurisdictions, many of them will not consider this as a GMO because we've not added a gene. So of the genome of tomato, I think around 3 billion base pairs, we've made one base pair change, one small knockout. In theory, you can make this change through breeding. It would just take many, many years to find that mutation. With CRISPR, we can do it very quickly and very efficiently, and actually we can multiplex, we can make many changes simultaneously. So CRISPR technology, to me, is an extension of breeding. It's just faster, and we can multiplex it. And therefore, this is a much shorter technology to come to market. Now, why are there not more genome-edited products on the market? Well, the challenge there is today we do not have a synchronized regulatory framework. This is a regulatory map of the world. So the pinky colors represent countries which have an established framework, a relatively low-cost, easy framework to launch a genome-edited product. In fact, this is slightly out of date because China now has developed a process. But what you see is Europe. In green here, they have indicated that genome-edited crops will not be GMO. However, they have not put the regulatory framework in place. So if you're a producer of genome-edited seed, like in soybean or other global traded commodities, that goes to Europe. Today, you will be fined for allowing that material to go to Europe. And this is preventing the widespread and rapid deployment of genome-edited crops around the world. The good news, however, is that a number of countries, such as India, China, and Japan, which have been pretty conservative on their regulatory framework, are now signaling that they're going to approve genome editing. In fact, some of these products, like in the healthy tomato, are now on the market in Japan because they realize the benefit of the technology and the relatively low risk. So with that, I'm going to pause, and Yuri and I will be open for questions.
Thank you both so much. We've got our first question in the Q&A, and I encourage other folks to go down to the bottom of the screen and type in any questions that you may have. First question comes from Pat Kina and says, why has there been no uptake of GMO crops in Russia? Who wants to take that one?
I think, you know, when it comes to specific countries, it's really a combination of the availability of the technology, the IP around it, the type of crops that are going to be used, and obviously any other political hurdles that can be associated with a certain technology. By the way, it's not just for GMO; it's for any technology, especially in the field of plant sciences. I'm not in a position to talk about the Russia and the policy of Russia for approving or disapproving, but you can imagine that the entire situation that we are facing. By the way, it's not just in research yet, but also before. Some countries maybe don't see that value, but I don't know, Ian, if there's anything you can comment on that.
So
Russia is primarily for wheat rather than corn. From a crop perspective, it's more complicated. Technically, it's just more complicated. But Ian, maybe you can build on that.
I can't make a comment specifically on Russia, but I can draw an analogy with China. So China has officially not accepted GM technology for many years. And in part, that was a political decision because they did not want to be reliant on what was perceived to be Western technology. So they did not want their food supply to be held hostage, perhaps, by a number of international companies. Now, China has invested enormously in plant sciences, both in GM and in genome editing, and they are now approving their first GM crops, 30 years behind the United States. But they are now adopting that technology. So, and that's because they've developed their own domestic industry for both GM and genome editing. So that may be the case for Russia. They don't want to be dependent on others.
Thank you. We've got one from Liviu in Romania. Do you see any signs of EU acceptance of gene-edited crops in the near future?
Yeah, I can take that one. Yes, well, it depends what you mean by the near future. There is a framework, there is a proposal that is going through the European regulatory system, and there's a draft document, which is actually pretty favorable to the technology in that it says you can make 20 changes of up to 20 base pair changes. So there is a proposal, and certainly Europe is, most European countries have not accepted GM with the exception of Portugal and Spain, but the EU are also banning a number of chemicals and even biologicals. So pharma groups and a lot of academics are getting very concerned that Europe is being anti-technology, and a lot of people are positioning genome editing as in part a solution to some of the challenges by withdrawing other technologies. So yes, there is a framework, it's being worked through the system. It is science-based, but it still has to go through that the whole regulatory process, approvals. Again, I'm not a regulatory expert, but my expectation it'll take a couple of years to finish that. What I do know is institutes and companies in Europe and global companies like us are developing products in our research pipelines for those markets, assuming that regulatory framework will be in place.
And the reality, and this is not about genome editing. I think it's more related to this whole presentation today. I don't think that we extracted, I'll be the last person to say that we extracted all the variation that exists in nature. But if we want to be fast, if we want to be precise, and if we want to be able to adhere to all the challenges that farmers are facing, growers are facing around the world, we have to look at it holistically and blend in technology into our ways of working. And that's basically what our functions are doing. I don't see a situation when in the future everything will be engineered.
Yeah.
You simply can't do that. It requires so much resources that is just not make it even economically viable for a company. We have so much of variation that exists out in nature. Our processes of plant breeding, as I mentioned in my last slide, using the data, the analytics, and being able to run models and select even sometimes on the computer of what type of genetic combinations or genetic fingerprint do we want, because it's high probability that it's going to work. That's nice and that's great, and it's going to continue to drive and foster our introduction of new varieties on the market, but sometimes it won't be enough. And unfortunately, the ones that pay the immediate price are not the Western world, you know, where you can get tomatoes probably 12 months out of the year, even if they don't grow in your nearest farm. Doesn't matter. You know, we live in the US, many of us live in Europe. You always have tomatoes on the supermarket. Tomatoes are grown in the summer. They need a lot of light. So either you are able to start growing it locally at a high cost that we can pay, or you're going to start importing. Other parts of the world, to be honest, we did the math, it's about 50% of the world population is being fed or relying on farmers that account for about, you know, 1/3 of the farmers worldwide. Vegetable farmers, I'm not talking field crop, I'm talking veg. 30% of 1/3 of the farmers worldwide are local, simple farmers that are dependent highly on the yield that is going to come from their field in order to, you know, provide food for their families. And they are developing the foods and producing the food for 50% of the world population. So just to put things in perspective, we got to bring those farmers along, so to make sure that they are more, first of all, profitable, but also more successful in feeding that growing population around the world. This is not just slogans. This is real events that we are facing, a real situation, an environment that we are facing today when we travel around the world and when we meet with those farmers.
I just want to pick up on a comment Yuri said there, which is about speed. I attended a crop modeling weather seminar that talked about predictions of crop yields in Eastern Europe. For certain crops, by 2050, they're expecting up to a 40% reduction in yield because of climatic conditions changing. And therefore, if our breeders, you know, are trying to breed for that situation, that's a huge challenge. And the breeding process, some crops, it can take 10, 15 years to breed a new crop. For GM technology, it can take 13 to 15 years. So speed is with those challenges of new diseases and climate conditions changing. Speed is a big issue, and that's one advantage of genome editing. When I first joined the company, I used a technique called tilling, which is a chemical mutagenesis of a population to create variation. It's random. I had to screen 120,000 seeds, one by one, planting them out. Took 2 years of me going to the greenhouse and testing them. If I did that with genome editing today, I could make 10 plants, and half of them, 10, not 120,000, 10, and half of them would have the edit I want. So, and I'm not saying editing is the solution to everything, but it's cheap and it's fast, and it can complement what our breeders do by adding in variation that can complement what's already in those elite varieties.
So we've got a question from Jason Jenkins here in the US, and he's saying solutions like BT traits have made a huge difference, but in many cases their effectiveness is breaking down. What are the next solutions coming through plant breeding?
Well, I'll take the BT question, and Yuri can sort of add to that with breeding. So again, insect resistance is a huge deal. BT technology has been very successful. However, both in South America and North America, we are seeing the pests evolving resistant. So it's kind of like antibiotics. In the end, the bugs figure out some way around that technology, and that's a huge deal, by the way, for fall armyworm in South America. Every other competitor trait for fall armyworm, the bugs have become resistant. The only technology on the market today is VIP3A. So one of the primary purposes of our research site here in North Carolina is to screen for the next generation of insect control. Not just us, but the other major competitors are all frantically working on next generation control for corn rootworm, for fall armyworm, and many other lepidopteran pests. So it's an arms race. You invent the technology, they become resistant, you have to come along with it. It's the same with the drug pipeline. Now, the good news is our technology, both in terms of AI-based protein design, high throughput screening, we have some more tools now. In fact, we just invested $18 million in a new insectary, which opened up at the end of last year to address that specific problem, to get the next generation of mode of action through insect control.
Yuri, what does that look like in the vegetable side? Are you finding similar challenges with this arms race between plants and pests?
For us, primarily on the viruses, and we have about every year and a half or 2 years, we have a new strain showing up on the market and posing some threat on the growers. Sometimes those strains disappear after 1 year. Sometimes they keep showing up year after year, kind of raising all the bells and whistles for us to start looking for potential resistance. Obviously, in veg, there is quite a higher sensitivity because it's fresh consumption of fresh products, sorry, which requires us to be more careful if you want. We still adhere to the same logic and the same kind of regulatory mechanisms. But in veg, for instance, today there are no GMOs. There were some attempts in the past, but today it's easy to do it, but it's just the regulatory process is just so expensive that it doesn't make it financial viable for us. So we are kind of constrained to those non-biotechnology processes. Ian mentioned some tilling. We do some mutagenesis in some cases where we need to, where we don't have the genetic variation. But this is where genome editing is kind of leaping over the GMO and providing us an opportunity, like you saw in the tomato case, to say, hey, we can join the race with this new technology and still adhere to the same regulatory and but also keep it more natural. If using the words from some people, can debate what natural is, about the way we develop our products. But we are mostly in a very conventional way of doing it. We and all the rest of the companies out there, simply because GMO leaves a very clear mark in the DNA, so it's very easy to identify if a plant has been modified, yes or no. So no one wants to go through that path, and it makes sense. If it's not, there is a resistance in the market, fine, we're not going to do that.
We have a question from Luis Pitombo in Brazil, and he's asking, what research with GMO is being done in the field in South America and Brazil? Current research are near the center of dispersal of local species. So what have you got going on down there? And then I'll ask you to expand that to what's happening in looking at, say, Africa, that 1/3 of those farmers that are feeding 50% of the population that you mentioned. Let's start with Brazil for Luis.
I can start with the Brazil question. So a number of our of the major projects we have running here in our RTP facility, I'd say about half of them are destined for the Brazil and Argentina market. So if we look at full armyworm, which I mentioned, both in corn and soybeans, other South American species in soybean, like soybean looper, velvet bean caterpillar, and then some of the diseases like Asian soy rust, which cause huge crop losses in soybean, particularly in Argentina, but also Brazil. So many of our projects, global projects, are targeted at those markets, and we work very closely with our colleagues in those countries to do the field testing. So we have to introduce the traits into relevant germplasm for Brazil and Argentina. We have to then test them in those field environments. So a very active program across Brazil. We also do work on the earlier research. So we work with agencies like Embrapa in Brazil to identify novel insect control traits, for example. So very active work, collaboration with the Brazil. In fact, in the past, I used to go to Brazil 6 times a year to work on GMO sugarcane, which is a very active collaboration with a number of institutes in Brazil. So a very key market for us.
Yeah, Yuri, do you have work on the vegetable and flower side in Brazil?
Again, yeah, obviously we're pretty big in Brazil and in Latin America in general, where fortunately vegetables is a prime portion of the diet of the individuals there. Again, we don't do any type of GMO work right now in veg. We believe that the genome editing could potentially bring us much more value going forward on certain traits, but certainly not everywhere. It just, as I said before, the amount of investment just doesn't add up compared to the current processes that we have.
Just to comment on the regulatory costs, there has been an initiative in the US I think many people realize that the GM crops are very over-regulated. So that when GMO came out in the 80s and 90s, there was a big concern about human health, environmental safety, and so on. And since that, so a very rigorous and expensive process was put in place. And as I pointed out earlier, that quote on 3 trillion meals and no ill effects, that came from an anti-GM protester who realized after 25 years, nothing bad had happened. So I think certainly in the US, there's been a move to streamline some of the regulatory processes between the different agencies, EPA, USDA, and FDA, to make it a more efficient process and allow technology to come forward.
I'm going to ask one last question just to follow up on that. And then I know we're at past the end of our hour. But how will advanced breeding, whether it's GM, gene editing, conventional, or any combination thereof, how will that get down to the level of farmers who at this point are not from the rich countries, are not broadacre farmers, but need that technology to continue to cope with climate change, feed that growing population, et cetera? That 1/3 that you had talked about earlier. What's that going to look like in the next medium horizon, I guess, or long term?
Ian, do you want to take that? Because I know you'll have a hard stop. So
I took a couple of comments there. First of all, GM crops are on the market in Indonesia, Bangladesh, India, Myanmar, Honduras, many of the developing world countries. I don't think it's enough. And particularly for Africa, there's been barriers and politics put in the way. So I would like to see the global regulatory framework for GM to be simplified. I think are more confident about genome editing. And I mentioned to you a number of the countries that were very anti-GM technology, like Japan and China initially. I've recognized that there are a lot of benefits of genome editing technology and minimal risks, because many of the things we create through genome editing, you could theoretically do by breeding, and that's largely a non-regulated process. So I think there's a big move by a lot of government agencies, academics, seed companies to try and get a simple, a synchronized regulatory process for genome edited crops. It is evolving. If I showed you that map a few years ago, it would not have looked as pink as it is today. I hope in 5 years' time we have a framework that can allow large companies, small companies, institutes to develop their own genome edited crops for their local environments, and let's not have the regulatory process be a blocker like it is today for GM.
Great. Thank you so much. And Ian, I know you've got to jump to another call, and Yuri, you've got one in a moment. So I want to just take the time to thank Syngenta, to thank Ian and Yuri for being here and staying after school with us even, and giving us a few extra minutes. And to encourage folks, we're going to do another live broadcast of this at 2 a.m. GMT. So you figure out what time that is where you are. But please encourage colleagues to check it out. We will put a recording up on our webinar page on our website. And again, huge thanks to Syngenta for bringing this great information to us and kicking off a series of webinars that we will enjoy in the coming year. So thanks everybody for being here, and have a great day.
Thank you,
Thanks for the opportunity. Bye-bye.
Thank you.
Thank you.